Dear everyone, welcome to Yokohama. I'm a LOC Chair, Harue Masuda. We have been worked hard to prepare this conference in these years and waited today to meet everyone coming to this conference. It is an honor to state the opening of Goldschmidt Conference 2016. As a representative of international and local organizing committee members, I appreciate everyone here that we can start the conference with your cooperation. This is the second Goldschmidt Conference held in Japan after 2003 at Kurashiki. At that time, we had about 1,300 delegates. This time, more than 3,500 delegates joined this meeting. During the history of the Goldschmidt Conference, started by the cooperation between Geochemical Society and European Association of Geochemistry since 1988, this conference has been grown up to be the most important academic meeting in the world for geochemists. We are also pleased that the Geochemical Society of Japan has been part of this honorable history as one of the organizing committees of societies. Yokohama is a rather young city in this country. However, it has been an important maritime city as a front door of Japan since our country opened to the world about 150 years ago. We hope that you will find charms of this city in addition to enjoy the science and encounters of old and new friends through the scientific sessions and official and unofficial events during the conference. And we wish all of you here will share the fruits of the conference and go back home with beautiful memories. Thank you very much.
Hello, on behalf of the Geochemical Society, I would like to join Harui in welcoming you to Yokohama and the 26th Goldschmidt Conference. I know that you're eager to hear the plenary talks. I'll be very brief right now and just give you 3 announcements. Just a reminder, please refrain from taking photographs during the sessions. Thank you very much for that. I also wanted to let you know that the exhibit hall will be open starting at noon every day, so you can go see the exhibitors and see the posters in a bit of calm before the poster sessions actually open at 5 o'clock. And finally, and most importantly, I wanted to remind you that we will have the Goldschmidt Medalist Lecture today at 1:45, given by the Goldschmidt recipient, Professor Alexandra Nowrotsky. So don't miss that. It'll be right here in this hall at 1:45 today. So with that, I'd like to welcome Professor Hiroyoshi Yuramoto, who will introduce our plenary speaker. Thank you.
Ladies and gentlemen, I am Hisayoshi Yurimoto, Chair of the Scientific Committee of this Goldschmidt Conference. I am delighted to introduce you Professor Satoshi Utsunomiya as today's plenary speaker. Utsunomiya-san received his PhD at the University of Tokyo in 2000. After his PhD, he worked in University of Michigan with Professor Rod Ewing, and Utsunomiya-san is now an associate professor at the Department of Chemistry of Kyushu University. From 2007. Utsunomiya-san is interested in application of advanced electron microscopy to environmental and geological materials to understand the principal mechanisms of various environmental issues. He has published more than 90 peer-reviewed papers and awarded a Fellow of Mineralogical Society of America in 2013. The title of his talk is Challenging Radionuclides in Environment at the Atomic Scale, Issues in Waste Disposal and Fukushima. Utsunomiya-san, please.
First, I'd like to thank session organizers and scientific committee members for giving me this opportunity. I'm giving a presentation focusing on two topics. First half of my talk is about some phenomena in near the nuclear waste disposal. And the second half of my talk is we'll focus on some remaining issues in Fukushima. So using the next 40 minutes or so, I try to convince you that small-scale phenomena is actually play a very important role to understand the fundamental processes that is happening at contaminated sites. As many of you know, when we operate a nuclear power plant, we produce this spent fuel. You see that the plutonium inventory in spent fuel is increasing every year. And when we reprocess the fuel, we have the high-level nuclear waste form that should be stored safely for more than 100,000 years. So, in general, we plan to store high-level nuclear waste form in the underground facility deeper than 300 meters with some artificial barriers. There are some processes that occurs near field, which I'm not going into detail in this talk. During the geological period, we will have to consider the failure of this artificial barrier system, and the radionuclides will be released and encounter the natural barrier, which is typical rock. In natural barrier, there are some large uncertainties remain in two factors. One is the nanoparticles. It can absorb the colloid, it can absorb the low-soluble radionuclides and facilitates the transport in the subsurface environment. Also, the microorganisms can immobilize the radionuclides and potentially retard the migration of radionuclides. They ubiquitously occur in natural barrier with great variety and a great amount. Let me start showing some research, example of the research on natural colloids. We worked on the plutonium transport, groundwater colloids in Mayak production association in Russia, and to evaluate the the effect of colloids on the plutonium transport. The Lake Kalachai is a source which was contaminated at the level of 7 times 10 to the 8th becquerel per liter. Our Russian colleague collected groundwater samples and conducted a variety of analytical techniques. Here you see the isotopic signature at 3 kilometers from the Lake Kalachai. It clearly reveals that isotopic, similar isotopic signature to the near field, and different from the contamination from the global fallout and accident. So this slide shows some composition of groundwater. Solodov and others reported the groundwater compositions near the source. And the only thing I want to point out is that there is no humic acid detectable in the groundwater. So that we don't have to consider the plutonium forming the complexes with humic acid. Basically, we evaluate the waste plume using the nitrate concentration. This slide shows the distribution of actinides to the different size colloid. 10 kD and 3 kD represents the 1.5 nanometers and 1.0 nanometers respectively. And a blue column shows soluble species after the filtration. So you can clearly see that the distribution of plutonium is different from the other actinides. The diagram on the right-hand side is a ratios of plutonium actinides bound to colloids and in solutions. You can see that the ratio is constant up to 2 kilometers or so, which may indicate that the apparent distribution to the major colloid phase that is responsible for the transport. And it also indicates that the colloid plays a very important role when it goes to the far field. The oxidation state of the plutonium is tetravalent, and the uranium was hit as hexavalent. The stability diagram of actinides shows that the uranium is probably possibly presented as uranium carbonate and plutonium as hydroxides. We observed a number of nanoparticles which are characterized as amorphous iron hydroxides, which shows a spherical shape with some impurities. The Nano-SIMS elemental map shows that the plutonium distribution is similar to the uranium distribution. This is an elemental map of one of these aggregates, and we can approximately determine that the plutonium-uranium ratio is about 0.03. So this was the first study that reported that was successful in imaging the plutonium association with specific colloids. Here I'm showing the other example of the colloid transport, which is in another test site. Some studies reported the plutonium association with natural colloid in a contaminated site. Among that, there's an important paper published by Dr. Casting and others, showing that plutonium was traveled 1.3 kilometers away from detonation point within 30 years period. However, the phase associated with these radionuclides were still unknown. So we had a chance to collaborate with Dr. Kirsten and obtain the groundwater colloid that exactly the same groundwater colloid that she used in this study. And we conducted TEM observation to characterize the radionuclides, some potential radionuclides associated with a colloid. So basically, we use the high transmission electron microscope. This is actually the hard stem imaging technique, in which the heavier elements shows a brighter contrast. So the cobalt is associated with some other metal compounds having the alvarite structure. And the rest elements present as the rest phosphate, which has a monazite structure. This may be the natural origin. We also found many uranium phases, species, like in sodium uranium silicate and uranium hydroxide. And among that, uranium and cesium phase were characterized. This phase can be stable at high oxygen fugacity and low cesium fugacity. And basically, in general, the cesium is considered to adsorb onto the zeolite in this system. This cesium uranate colloid is another pathway of cesium transport in the underground, in the subsurface environment. To summarize this part, the nanoscale colloids play a key role on the plutonium transport through the subsurface in Mayak site. The plutonium, 70 to 90% of plutonium is bound to the colloids. In case of Mayak, amorphous ion hydroxide is an important carrier of plutonium. In the oxidizing conditions, plutonium and uranium can be distributed in a similar manner, which may be the kind of important aspects. And different colloid phase can be responsible for the migration of each radionuclide. So moving on to the topics, radionuclide interaction with microorganisms. It's well known that there are two mechanisms of radionuclide immobilization by the microorganisms. One is the metabolism-independent processes. The radionuclides bound to the ligands on the cell surfaces, and the metabolism-dependent processes. The radionuclides precipitate and mineralize, by which the microorganisms can get energy. So we try to understand the post-adsorption process of rare elements, including mechanism and kinetics and speciation, during a long-term exposure using model microorganisms. We use the Saccharomyces cerevisiae, which is yeast. We use the rare elements as a surrogate of trivalent actinides. We conducted experiments in collaboration with the Japan Atomic Energy Agency, and we have done some work, series of work using this technique. And we conducted kind of simple experiments. Saccharomyces cerevisiae, we harvested Saccharomyces cerevisiae, and we took them out and we released it in an NaCl solution. And the rare elements solution with 0.1 millimolar is added to the Saccharomyces cerevisiae, and the solution pH was adjusted to the 3 to 4.5. And after a certain period of incubation, we took out the solution and solid for the further analysis. The important point here is we did not add the phosphorus in the solution to see the process of phosphorus released by the microorganisms. Here you see the time course of cerium concentration in solution. You can see that the cerium concentration faster decrease at the higher pH. When there is no cerium addition in solution, we observe the phosphorus release as a function of time, as well as organic matters. And the 80% of phosphorus is in also phosphate. A large number of cerium phosphate nanoparticles precipitate on the cell surfaces, as you can see here. And we can confirm that the precipitation on the cell surfaces by 3D tomography and TEM. And this cerium is identified, oxidation state of cerium was trivalent. And the structure of this rare earth phosphate, cerium phosphate, was identified as hydrous rare earth phosphate, which is Raoul-Fan. When we change, we also observed that the property of rare earth phosphate changes when we change the rare earth element. Here you see that the ytterbium phosphate nanoparticles precipitated on the surfaces, and these nanoparticles did not show any diffraction maxima, which means that it's amorphous structure. We also found that these nanoparticles survive after a long period of exposure. We did further experiments to get evidence of the post-adsorption mineralization processes. First experiments, the rare phosphate element was added to adsorbed to the yeast cells surfaces and removed these cells and put it in sodium chloride solutions without any phosphorus. And then after 7 days of exposure, we found the rare earth phosphate nanocrystallization on the cell surfaces. We also tested these similar experiments on the abiotic system using the LN resin and CMC, which are the representatives of the phosphoryl and carboxyl group. We didn't find any formation, the ytterbium phosphate formation on the surface of these resin. After that, some kind of a strange idea came up to my mind, and what is the impact of interaction of nanoparticles with microorganisms? So at that time, we already knew that there's some process. We already knew that there's some processes that can happen between the interaction between the nanoparticles and microorganisms, such as toxicity. We However, little is known for the effects on the metabolism. So we try to understand the process in the interaction between the microorganisms and nanoparticles by using the low-soluble cerium oxide nanoparticles and commonly used Saccharomyces cerevisiae. To make a long story short, we didn't find any cytotoxicity. We also we found the increase and decrease of some specific organic matters of extracellular polymeric substances when we put cerium in the system. And also, when we look at the internal substances, the two-dimensional the electrophoresis clearly reveals some expression and depression of some proteins. Among that, ENO2 is involved in the glycolytic system. So that this is evidence that the interaction with nanoparticle alter the metabolic processes, leading to the changes in the released substances. So to summarize this part, we have demonstrated that the inside compound and from microorganisms to form the nanocrystallization. This actually nanocrystallization can have an impact on the metabolisms of the microorganisms. I didn't show any data, but the released substances also in turn the effects on the colloid stability. One of my students will give a presentation tomorrow at the poster session on this topic. So the second half of my talk, we are showing some examples, results, remaining results. Sorry, the result we took in Fukushima. You can see that there is a contamination of the surface environment near the Fukushima Daiichi nuclear power plant, and the many Japanese scientists worked very hard to contribute to the better understanding the effects of the radionuclides. Not only me, but also the other Japanese people, scientists. So just to recall the amount of radionuclides released from the Fukushima Daiichi nuclear power plant has already reported in 2011. And among these radionuclides, cesium-134 and 37 are of great importance at the present. And based on this radioactivity, we can calculate the total mass of the cesium to be the 5 kilogram. Previous studies reported that cesium soluble form of cesium deposited on the surface soil and remains at the top 5 centimeters of the particle profile. And the cesium can be absorbed onto the clay minerals with forming the inner sphere complexes. And some TEM observation has confirmed that the ion exchange processes. And also, we know that Frederick site has the highest affinity to the cesium. In collaboration with JEA, we also investigated the contaminated area near the Fukushima. As you see that a lot of the cesium is fixed at the top 5 centimeters of the particle profile. Following the previous study, we also performed the sequential extraction method to separate the water-soluble, ion-exchangeable, carbonate metal, cesium bound to metal oxide, and cesium bound to organic matter, and bound to residue. Here you see that the size distribution of cesium radioactivity as a function of on the soil, in the soil. On the horizontal axis, we have particle size, and the radioactivity on the vertical axis. As you can see that the greatest amount of cesium activity is in submicron sized particles. And the sequential extraction of submicron sized particles shows the highest the amount bound to the residue fraction. This residue fraction is actually the submicron sized particles has radioactivity, and these radioactive particles consist of silicon and aluminum and iron and potassium We also found the interesting platy shape estuary sediment in the Puma River area. So the cesium, it shows, autoradiography shows the cesium bearing particles concentrated in the bottom part and the gradual increase of the number of the particles towards the top, which means that the cesium, number of the cesium bearing particles changes over a few years of period. And this sequence extraction shows that 89% of contribution to the residue. So this, the estuary sediments, when we tested the artificial desorption experiments in artificial seawater, we've got 3.4% of cesium dissolved in so it means that it can go into the ocean, and only 3% of radioactive cesium can be dissolved in this ocean. So this is a kind of old model that I proposed a couple of years ago, and so the cesium deposited on the surface soil and bound to the submicron-sized particles of clay minerals, and the behavior of clay colloids governs the migration and distribution of cesium in subsurface environment. So which is probably still true, but but when we look at the soil samples, we started finding a very hot spot, very highly radioactive spots in soil. As you can see here, here and here. We can also see that hot spots even in estuary samples. We also got the air filter sample from Tokyo, and autography clearly shows a numerous amount of hot spots. So this is collected on March 15th. Just to remind you that the integrated annual dose for the internal exposure was just estimated to be 50 times less than the regulatory limit in this case. So just to recall where they came from, we collected sample from very close to the FDNPP, and Tokyo is actually the 230 kilometers distance from the FDNPP. Some very interesting article were published by Adachi and others showing the cesium-rich microparticles. They're discovered 170 kilometers distance from the Fukushima nuclear power plant, and they contain silica and iron and zinc and cesium as major elements, and they are amorphous and insoluble in water. So I thought that maybe these hot spots we found in the soil is attributed to these cesium microparticles. And we think that the cesium microparticles retain the valuable information that investigating this cesium microparticle can solve some chemical form and release process of cesium and radionuclides, and chemical form inside chemical reaction inside primary containment vessels. So the aim of this part was to further understand further details property of cesium microparticles and chemical reactions during the meltdown, and the contribution to the radioactivity evidence and evidence of migration of cesium microparticles in the surface environment. So we picked up these hotspots by repeating the autoradiography many times, and we searched this cesium particle using SEM, and after that we measured the gamma radiation by gamma spectroscopy, and after that we made a thin foil using the FIB for the TEM analysis. So these are this slide shows the SEM image of the cesium microparticles found in Fukushima soils and river sediment here. So you can see that the size of the particles are less than 5 microns, and however, the radioactivity goes up to 279 becquerel. They are actually sometimes form the aggregates in Otozawa, very close to the FDNPP. And 7 cesium microparticles were detected in the air filter in Tokyo. The size is about 1 micrometers, and the radioactivity ranges from 0.1 to 2 becquerel. Here you see that the high-def-STEM image of cross-section of 4 particles. You can see that the SAED pattern shows almost amorphous. And based on the STEM ADX analysis, major elements are oxygen, silicon, iron, zinc, and cesium, as reported before, and with some minor elements like this. And cesium concentration is as high as 36% in case of this particle. And this particle looks very interesting texture, like it shows large pores inside. And another particle from the Fukushima sediment from Koirino surface soil. We found also the large pores and also the stick-like inclusions here. And the distribution, elemental distribution of inside this particle shows almost homogeneous, however, except for the cesium distribution. This SAED shows almost amorphous pattern. And the electron energy loss spectroscopy revealed that this silicon is presented as silicon dioxide, not metallic silicon. The cesium, iron is also exists as iron oxide, and barium it was derived from the beta decay of cesium. And also, we didn't find any boron, which indicates that there's no volatilization of boron and carbide in this case. These are the elemental map of some other particles. Showing some homogeneous distribution of elements, except for the cesium. And also, we found some inclusions in there. Although it appeared as homogeneous, we found a number of nanoparticles, a large number of nanoparticles, when we observed at nanoscale. As you can see here in high-def-STEM images. There are a lot of heterogeneity. And the elemental map shows that these nanoparticles consist of zinc and iron, mainly, and associated with zinc, tin, cesium, and chlorine. Dark area is composed only of silicon. So based on the TEM, high-resolution TEM and FFT images, we were able to characterize this zinc iron oxide as franklinite nanoparticles. So that we can tell that this cesium microparticle is mainly composed of silicon dioxide glass and zinc iron oxide nanoparticle aggregates. And cesium and tin simply associated with the franklinite nanoparticles. This slide shows the fission product inclusions. We sometimes find zinc silver telluride, and also even selenide we found. And here we found a cesium chloride in one pore. The sticky particles we found in the Koirino sediments were further characterized by TEM. In the TEM, a small portion remained as crystalline, which we were able to measure the despacing as 0.39 nanometers, which is corresponding to the 004 plane of cesium hydroxide. This is the first identification of the cesium hydroxide. And importantly, we found uranium in this area. There are many the Franklin atom particles. A few of them are associated with relatively high concentration of uranium. We didn't find any uraninite. We found uraninite is simply associated with zinc ion nanoparticles. This means that indicates that the uraninite was partially vaporized, volatilized. However, it is well known that the uranium is classified as non-volatile elements. So we think that some of the uraninite was oxidized a little bit, and it can be partially volatilized at relatively low temperature. This slide summarizes the composition of cesium microparticles. Linear correlation between there is a linear correlation between the iron and zinc versus silicon contents, even in the point analysis, both in the point analysis and error analysis, which means that this particles is composed of silicon dioxide and franklinite nanoparticles, and the number of the franklinite nanoparticles constrains the concentration of the iron and zinc. And the cesium concentration appears to correlate with silicon dioxide. However, when we plotted the point analysis, we didn't find any correlation, meaning that cesium is kind of adsorbed onto the surface of the franklinite particles at various concentration. We think that these cesium microparticles are produced by the molten core concrete interactions. Because MCCI is consistent with the texture we found so far, there are many pores which may be ascribed to the degassing and the vaporization. Some people may wonder why these particles didn't contain the composition of the concrete. This is simply because the volatilization temperature is much higher than the temperature of reducing reaction of the silicon dioxide. So we are proposing the possible scenario during the meltdown. During the meltdown, some fission products are released and form the nanoparticles. And during the meltdown, the failure of the pressurized reactor pressure vessel, the zinc ion oxide nanoparticle form, and some cesium species absorb onto the surface. And then when the melted core hits the concrete, the silicon dioxide, silicon monoxide was generated, immediately condense to form the silicon dioxide on the aggregates of the zinc ion oxide nano aggregates. During that time, it can incorporate some fission product nanoparticles. In order to understand the contribution of cesium microparticle to total cesium radioactivity, we conducted the dissolution experiments of Tokyo air filter using the ultra-pure water. This autograph shows before and after. They are almost identical. So we actually immerse the filter in a solution with static conditions, so that the cesium microparticles do not resuspend in the solution. Anyway, After this experiment, we found surprising results. The insoluble fraction of which corresponds to the cesium microparticle fraction can occupy the 89%, as high as 89% of the total cesium radioactivity. So because we found the cesium microparticle in various environments, we can conclude that the cesium microparticle can be another important carrier of cesium pathway, media of cesium migration in this surface environment. Should be taken into account. In conclusion, we for the first time characterized cesium species using TEM, and we also proposed a possible reaction processes inside reactors. We are proposing the model of cesium migration, including a contribution of cesium microparticles in the surface environment. Cesium microparticles was a dominant contributor to the total cesium radioactivity during the highest radioactive fallout event in Tokyo. So I'd like to highlight that cesium microparticles are another important route of the dispersion of fission products and actinides, such as we found in uranium, in the environment. Cesium microparticle can provide valuable information on the condition inside the reactor. Lastly, there's still the biggest issue remaining in Fukushima, which is decommissioning. We have to get the information on the melted spent fuel, melted core, as much as possible before removing it. We think that the cesium microparticle can have potential to provide such information. It is tiny, but it can tell some important information. Finally, I'd like to thank some funding sources, and I'd like to thank many collaborators here. And also, I'd like to thank my families, especially for my wife. She always supported me until last year. And I would like to thank my group members in my group. Without their help, I could not continue the research for this last year. And thank you for your attention.
Thank you very much. Very fine scientific result and very shocking result. So, question? Yes?
Thank you very much. So we have a more sensitive way to measure radioactive cesium isotopes, like 134, 137, more sensitive than the existing methods. And my question is whether you need such a sensitive way. Are the existing methods good enough for your application?
Well, I think at this point, I want to say that the existing method is enough for this kind of work. However, some other Japanese scientists try to understand the behavior of cesium in environment, which sometimes at very low concentration. So those scientists may need your techniques to analyze cesium.
So other question? Yes, please. Please use the microphone.
In your conclusion, you showed a very elegant experiment showing that 80–90% of the cesium was associated with the microparticles, and that was insoluble. Mm. But when I think of the soil samples at the beginning, you said it was associated with the clay.
Right.
So that conclusion is only for the certain filters that you caught with fallout? Or if you look in that estuarine sediment, would you say what fraction of the cesium is with microparticles in that estuarine environmental sediment?
That's very good question. The reason why I only showed the result of the air filter is that we can only estimate the contribution for the air filters, because we already have cesium adsorbed onto the soil minerals, so we cannot get a contribution, estimate the contribution of cesium microparticle when we analyze the soil samples. So, but some of my students trying to develop such the method to get a contribution of cesium microparticle from the soil samples right now. So,
yes, please.
Hi, very interesting talk. I was curious if these microparticles that are rich in cesium exist anywhere else, and if they can form in any other way other than, obviously, the Fukushima tragedy. In other words, what are the controls?
What do you mean, control?
Did you look at other environments, and did you find similar particles? Can they form in other ways?
In other ways? You mean Chernobyl or something?
Other mechanisms. Other than
other events.
Nuclear disaster.
Nuclear disaster. Well, that's very interesting question. And I know that some people reported this hot particle for the Chernobyl accident, and I haven't seen any report from the TMI to accidents. So these are probably the limited number of studies of cesium microparticles, I think. But it can form when you get MCCI interaction. Molten core and concrete interaction. So you have to have a melted core hit the concrete pedestal of the primary containment vessel. But we don't want such a severe accident, you know.
So any other question, comment, suggestion? Yes, please.
Thank you for your talk. I work on cesium bioavailability from the sediment. So I want to ask you how we mobilized this cesium bound to the microparticle. Is it any other process that easily remobilize this binding, that this cesium can be bioavailable to plants or other organisms?
You mean bioavailability of this cesium microparticle, you mean?
How strong is the binding of cesium to these microparticles? Are any other process would lead to the dissociation with the particles?
You mean that the bioavailability of the cesium microparticles in environment?
Actually, this is two questions. The first question is, you have shown that 80 to 89% of the cesium radioactivity is on these microparticles.
Right.
So my first question is, how strong is this binding?
The binding.
The binding.
Okay, so the cesium is, as I showed you, the cesium is associated with the Franklin-Knight nanoparticle, which is incorporated in the glass, silicon dioxide glass. So it's insoluble, I would say.
Okay.
So the SiO2 glass has to dissolve to release the cesium in this case.
So in this case, you mean that most of the cesium bind to it is not soluble after the binding. Is that correct?
You mean?
You mean it's insoluble?
It's insoluble, yeah.
Okay, I see. Okay, thanks.
Yeah, you're welcome.
So any other question, comment, and suggestion?
Hello. My question is, how often did you find the cesium particles?
Excuse me, where is
Here.
Yes, over there.
Huh?
My question is, how often did you find the cesium-containing particles in your samples?
Well, I took some hint. Like, it took 4 months to discover the first cesium microparticles for my students, But after learning how to detect the cesium microparticle, he kind of get used to these techniques. So he can probably detect and find the cesium microparticle maybe a couple of particles per month or so. I'm not sure, but there are many cesium microparticles, as I showed in autoradiography.
Yeah, okay, time is up. So thank you again to the speaker.